I want to thank everyone for joining us, all of our attendees from around the world, and a particular thank you to our speakers, again, from all around the world. Technology is fantastic. It's wonderful what we can do with Zoom and put these meetings together at a moment's notice. Science is moving quickly, and it really is wonderful. So as you all know, taste and smell are incredibly important, and COVID-19 pandemic just reinforces how important these essential senses are. We're going to hear a lot in our symposium from the several speakers on the specific details, but hopefully this will give us some insight into the disease, diagnosis of COVID-19, because loss of smell and potentially taste are key diagnostic symptoms of the disease. And hopefully this will also give us insight into the underlying biology of the disease and of the relevant taste and smell cells. So with no further delay, let me turn it over to my colleague, Pam Dalton, who is going to give another few introductory remarks. Thank you.
Well, thank you. I wanted to be able to share my screen here just to give us a little bit of background on what we're going to hear today. Is that this pandemic has shone a light on the chemosensory systems like nothing in my 25 years of research has done before. And while that is both enormously important, it's also going to provide a significant burden on people for whom these symptoms occur and they do not recover. So I just wanted to give a little background. What we're talking about is olfactory dysfunctions that fall into a couple of different categories, and these are particularly relevant for post-viral olfactory loss, where you can either become completely qualitatively unable to smell or somewhat impaired by anosmia, which is total loss of the ability to smell, or hyposmia, which is some degradation of your ability to smell, as well as qualitative changes in the ability to smell, such as phantosmia, which is smelling things when there is no apparent chemical producing a input to the olfactory system, or parosmia, where things that you can actually detect smell qualitatively different and usually far more unpleasant than the indicator, the parent molecule. I'm actually trying to see how I can change my slide. Okay, there we go. Next. So there are two kinds of dysfunctions that occur in olfaction. One is conductive. It has to do with either mucus congestion or edema in the mucociliary glands that prevent the molecules from actually reaching the olfactory receptors. And this is the one that's most common to all of us when we have a standard cold, where we start to get mucus congestion and we stop smelling, or we think we've lost our sense of taste or flavor because the molecules that either arrive through the ortho or the retronasal pathway can no longer reach the receptors. And because our noses are blocked, we don't normally concern ourselves with this. But often this kind of blockage is not the only reason why someone loses their sense of smell. There can also be changes due to non-conductive factors that actually the virus particles or the viral itself or something else targets the olfactory receptors themselves, or maybe the sensory neural system at a higher pathway. So these two ways of actually understanding changes in olfaction when we get something like an upper respiratory infection are widely understood, but what is different now in this current pandemic is that this doesn't seem to, the conductive changes do not seem to be part of it. And so people are losing their ability to smell or taste or experience chemical senses through these chemosensory systems abruptly without the concomitant conductive congestive changes that would allow them to experience it and not worry about it. It turns out that in terms of olfactory dysfunction, upper respiratory infections are the second most prevalent cause, followed by sinonasal disease. The difference with sinonasal disease, however, is that sinonasal disease can be treated. You can have polyps that are surgically or pharmacologically changed and intervened. You can have steroid treatments, for example, where it fluctuates from time to time. Upper respiratory infection is very common. However, fortunately, most people, I would say the evidence is between 80% and 85% of the people who experience an upper respiratory loss actually do recover some or complete or partial or complete olfaction later. But it's still the second most prevalent way that our olfactory system is attacked. Unfortunately, there can be some individuals, and we still don't understand all of the reasons and all of the factors why there is complete recovery from the URI, the congestion goes away, and some individuals never recover olfactory function or normal olfactory function. And so as a consequence, this pandemic has given us an opportunity to not only shine a bright light on the importance of the chemical senses, which I will relate a small anecdote to illustrate. A long colleague of mine at the Monell Center had an upper respiratory infection a few years ago, and like most people, lost his sense of smell. But because he was so close to the chemosensory community and understood the consequences of this, actually told me that he experienced significant anxiety and tested himself regularly on multiple days until it returned to normal, which fortunately for him it did. But that's just the consequence of this when people don't understand what the loss of chemosensory ability provides. There are huge consequences. It relates to safety in the world that we don't always understand, that we are constantly monitoring our environment for things like natural gas leaks or even cleaner solvents that we use in our home that might become at an unacceptable concentration. Food-related changes. Some of these go into the point where people overeat, trying to enjoy food the way they once did, or not eating at all and losing weight. Both of those are not good conditions. Social conditions where people worry about their own body odors, or they can no longer enjoy the odors of the people that they love. That's the smell of a loved one, a husband, a child, a newborn baby, and this is very important. And those consequences go throughout life and produce often an anhedonia, a loss of hedonic living, a loss of enjoyment of life that can't be qualified or quantified in tests or changes, but actually have a huge impact. And so while this pandemic has shown us this bright light on the importance of these chemosensory systems, I think what is more important, it's also given all of us researchers an opportunity to learn a great deal more about the pathophysiological of the loss, its prevalence, the risk factors that may underlie who recovers from olfactory loss following URIs. And the speakers that have been assembled today are leaders in the community who are able to address all of these factors. And I'm really excited to hear from all of them, and I hope you all are also. Thank you.
Pam, thank you very much. So I'd like to welcome our next speaker. We're very pleased to have her. She comes to us from King College London. So go ahead.
Thank you very much. So I'll just... Okay. So thank you very much for inviting me to present here today. So I'm going to present some work we've been doing on the symptom study app, which was developed in order to track symptoms over time for the COVID-19 epidemic in the UK, US, and Sweden. And the results I'm presenting have actually been published as a bit of communication in Nature Medicine. So since the outbreak of the COVID-19 infection in China in December, there have now been more than 6 million cases worldwide. And although many people have flu-like symptoms, there is still in most countries not inadequate massive population testing. And so it's very important to identify the combination of symptoms as most predictive of COVID-19 in order to guide recommendation for self-isolation and also prevent further spread of the disease. And so the aim of our study was to investigate whether loss of smell and taste that had been reported in the small case reports and the media is specific to COVID-19, and we included 2.6 million users of this COVID symptom study app. And then the second aim was to identify the combination of symptoms that are most predicted of a COVID-19 infection. So the COVID symptom study app was developed by Zoe, which is a computer-based company, in collaboration with King's College London and the Massachusetts General Hospital. And it collects data for from both asymptomatic and symptomatic individuals, and it allows to track how the disease is progressing over time, because it records self-reported health information on symptoms, hospitalization, comorbidities, demographic characteristics, as well as PCR test outcomes. And so these results are based on the 21st of April data freeze. So we had 2.4 million UK users and about 168,000 US users. In the States, the app was launched a bit later. So out of those 2.5 million, we first included only people that reported having had a PCR test and having received an outcome of the test. And those amounts to about 15,000 UK users and 2,700 US users. And what we found was that among those tested positive, loss of smell and taste was threefold higher than among those tested negative. Suggesting that people with loss of smell and taste should self-isolate. So in this same data set, we then looked at the association between other COVID-19 symptoms and COVID-19. And here I'm presenting the odds ratio and the 95% confidence interval in blue for the UK and in orange for the US. And as you can see, the odds ratio for loss of taste, smell and taste is huge. It's 6.5 in the UK data and over 10 in the US data. But also we identified other symptoms which are strongly associated to being tested positive for COVID-19, including skip meals, fatigue, fever, and persistent cough. And not all results were replicated in the US data, but this is most likely to the smaller sample size. So, so then we wanted to look at the linear combination of symptoms most likely to predict COVID-19, being tested positive for COVID-19. And so we randomly divided the UK set into two subsets, and we performed a stepwise linear regression to identify independent symptoms adjusting for age, sex, and BMI. And we found besides age and sex, the most predicted symptoms were loss of smell and taste, severe or significant persistent cough, severe fatigue, and skip meals. And then we tested our model both in the UK test sample and we validated it in the US sample. And as you can see, the model is quite good. It has an area under the curve of 71% in the UK sample and 76% in the US sample. And the sensitivity and specificity are 65 and 70% respectively in the UK, and quite consistent in the US data. And then what we did, we applied this, this model to the data from all the app users who reported symptoms. And there were, out of the 2.6 million, 800,000, slightly more than 800,000 reported some symptoms. And we predicted that out of those, 17.42% were likely to have COVID-19 based on our models. And this is, sorry, smaller than this. So this is including, it wasn't published as part of the Nature Medicine paper, but it's now in press in Lancet, and it's basically showing, is a Venn diagram showing the overlap between individuals reporting having loss of smell and taste, persistent cough or fever. In the UK at the moment, only these three symptoms are in the COVID-19 symptom list. So this data is based on the data feeds from last week that includes 13,000 tested positive cases. And as you can see, 16% of people that are positive for COVID-19 report having loss of smell and taste, but do not have fever or cough. And so having added loss of smell and taste to the COVID-19 symptom list is perhaps allowing to discover many more cases that we would have just having fever or cough. So our study has some limitation, which first of all is the self-reporting nature of the data. So this is prone to information bias. Also, our app asks just one question which combines loss of smell and taste. So we're not able to tell whether the person has just one of the two symptoms. So results are likely to be conflated. And then, as was discussed before, our app users are not representative of this general population. So we are missing people belonging to the older age group or to ethnic minority or people living in more deprived area. So we do have a selection bias. However, we do find the loss of smell and taste is a strong predictor, potential strong predictor of COVID-19. And the COVID-19 does appear to cause problems of smell receptors in line with many other respiratory viruses. Based on the Venn diagram I showed you, but we believe that perhaps screening tests should not just be focusing on measuring fever and so on temperature sensors, but should perhaps melt the different screening tests should be introduced in workplace and entrances, and they may capture a greater number of positive cases. And we identify a combination of symptoms that includes anosmia, fatigue, persistent cough, and loss of appetite that together might identify individuals with COVID-19. So I just want to thank people in my department. This is Zoe, and these are the twins that are part of our studies. Thank you.
Cristina, thank you so much. We really appreciate you coming by today. Our next speaker is Liz Cirulli of Helix. So Liz, can you go ahead and share your screen, please? And while we're waiting for Liz's to put her screen up, let's go ahead and just direct our attention to the question and answer panel. So if you have questions for our speakers, please drop them in the question and answer panel. And so we'll go ahead and hear from Liz. Liz, go ahead.
All right, my screen's up good?
Yeah, you look great. Go ahead.
Fantastic. All right. So my name is Liz Cirulli. I'm a scientist at Helix, and I'll be talking about some of the work we're doing with COVID-19 research. So a big goal of the human genetics community right now is to identify genetic variants that influence the differences between people and how they respond to infection with SARS-CoV-2. As we all know, there's a lot of variation in how people respond, from people having no symptoms at all to people losing their lives to this illness. And a lot of the risk factors have been characterized, so older age, male sex, having comorbidities, but there will be genetic differences that influence the way people respond as well. And so the human genetics community is really coming together actually in a way that's been really great to see, to try to all pool our data and try to make these discoveries to identify what's causing some of these differences. So how is Helix contributing to this effort? Helix is an Exome sequencing company, and we perform Exome Plus sequencing. This means that for everybody that we sequence, we produce a clinical grade exome, which means that we're sequencing the exome and getting all of the rare and even unique variants in the protein coding portions of each person's genome. Even if it's a mutation that's unique to them, the exome sequencing will get it. Our sequencing assay also produces the equivalent of a GWAS backbone, which is basically equivalent to doing a GWAS genotyping chip. So we can impute out from that and do any of the basic GWAS that you would do with common variants throughout the genome with any other GWAS method. We have sequenced hundreds of thousands of individuals with this assay, and we have a core set of a little over 200,000 unrelated individuals that we have been using for different research projects. What we did for this project was we contacted individuals who we have sequenced who had provided consent to participate in research projects, and we sent them a COVID-19 survey. So this survey has now been responded by about 14,000 people. It's a 10-minute survey that's based on kind of standardized questions that have been being collected by one of the worldwide efforts that's looking at COVID-19 host genetics, named the COVID-19 Host Genetics Initiative. So it's a 10-minute survey. It's pretty comprehensive. It collects information about symptoms, exposures, comorbidities, medications, demographics. We're doing longitudinal follow-up, so we're getting a lot of good information from these people. And everybody already has their exome sequenced, so we're able to do genetic studies right away as soon as the data come in. We've performed different genetic analyses already and have submitted them to the COVID-19 Host Genetics Initiative, which is pooling together genetic results from multiple different groups throughout the world. And those meta-analysis results are available on their website. I'm not going to talk about genetic studies today because unfortunately nothing statistically significant has been found yet from any of these studies. So we're all still trying to build up sample sizes and figure out the best way to do this. But we do have a lot of interesting information from the phenotypes we've been studying, and so I would like to talk about that. So here's a brief overview of our some of the information we get from our survey. So we've had about 17% of respondents report that they've been exposed to someone with a COVID-19 diagnosis. 12% of respondents experienced a fever during the pandemic period, and 6% experienced those kind of classic COVID-19 symptoms of the fever, dry cough, and loss of sense of taste or smell. 3% of the respondents had COVID-19 tests, and 10% of those tests were positive. Because not everyone has a COVID-19 test, we're of course interested in identifying the symptoms that are associated with having a positive COVID-19 test in much the same way that Cristina Menni just talked about. So because if you can identify who is more likely to have COVID-19 from their symptoms, then that has medical implications, and that also can allow you to do better research studies by improving your power. So shown here are the 29 symptoms that we queried our individuals about, ranked in order from left to right in terms of how enriched those symptoms were in people with a positive COVID-19 test. The yellow line is the people who is the proportion of people with a positive COVID-19 test who reported each symptom. The pink line is the people with a negative test, and then the dark blue line is the people who weren't tested. So you'll see many of the well-known symptoms over on the right as enriched in people who had a positive COVID-19 test, including loss of sense of taste and smell. And then all the way on the right, you'll see sensitive skin, which is a novel phenotype that hadn't been queried very much before, but that we added to our survey because of some firsthand accounts that we had heard. And we find it to be quite enriched in people with a positive COVID-19 test. We actually went ahead and used one of the models that was put forward by Cristina Menni, Tim Spector, and others on our cohort to see how well it performed in our group. So the model that was originally presented is shown on the left, and we applied it to our data on the right. We get an area under the curve of 0.8 for that model, so it works very well in our data set as well. If we add skin sensitivity into the model, then it performs a little bit better. And if we try a few other models, something like just anyone who's had a fever, loss of sense of taste or smell, or skin sensitivity together, then that also produces a pretty good model. So it's great to see different groups, especially with large sample sizes, collecting information like this and being able to model who is likely to be a positive COVID-19 case. One thing that is coming up, though, as time is going on and as we're getting more data in, and especially longitudinal data, is we're seeing that the way that these models will work will be influenced by a number of different factors that need to be taken into account when doing analyses. So one of the things that I've noticed is that the symptoms that are enriched in people with positive COVID-19 tests are also symptoms that are just enriched in people in general. That are enriched in people who are sicker in general. So what's shown here is the mean number of symptoms that are reported per person in our cohort. So if you look at all respondents, there were just over 3 symptoms reported per person on average, just everyone who took this questionnaire. If you look at people who reported at least 1 symptom, then it gets a little bit higher than that. And then if you look at the mean number of symptoms that are reported by people who report having sensitive skin or who report having a loss of sense of taste or smell, now you have more like 10 or 12 symptoms being reported per person. If you then layer onto this graph, here I add 2 more bars on the bottom, showing the mean number of symptoms in people who had a positive COVID-19 test versus people who had a negative COVID-19 test. And you can see here that the people who had a positive COVID-19 test had on average 10 symptoms that they reported. And the people who had a negative COVID-19 test had almost 6, a little less than 6 symptoms per person, and this was after restricting to people who reported at least 1 symptom. So all of these people who had a negative COVID-19 test, they did at least report being sick in some way, but they still had fewer symptoms than the people who had positive test results. This gets perhaps even more interesting if you look at it kind of spread out over time. So we've collected a couple of different tranches of data from individuals at this point. And so we basically have split it here into two groups. The data set that we collected in late April, which reflects information about people's illnesses during February, March, and April. And then a data set collected in late May, which catches information reported by people in May and in late April as well. Shown on the right is the mean number of symptoms per person who had either a positive or a negative COVID-19 test result during these two time periods. And you can see that earlier on, the people who were getting COVID-19 tests were more sick than the people who are getting COVID-19 tests nowadays, which makes sense. There was a testing shortage at first, especially in the US where these data are from. And so only the sickest people were able to get a COVID-19 test, and now it's much more widespread. And so people are able to get tested for COVID-19, even if they don't have as many symptoms. What's interesting here, though, is that this decrease in the number of symptoms is also seen if you're comparing positive cases to each other, as well as if you're comparing negative cases to each other. So in both time periods, the proportion of the number of symptoms reported by positive cases was higher than the number of symptoms reported in people with negative results, and those proportions went down in both cases, both for the positives and the negatives with the more recent data. And this also goes along. I'll point out on the left here that about 15% of the test results were positive in the first data set, whereas only 6% of the tests were positive in the second data set. So more people are getting tested and fewer people are getting positive COVID-19 tests. Another interesting thing to notice from this is that the proportion of people who have these kind of classic COVID-19 symptoms amongst the people with positive test results is also seems to be decreasing over time. So again, it's split into the earlier test results and the later test results here. And now shown is the percentage of people who report either loss of sense of taste or smell or report sensitive skin, split up into people who had a positive or a negative test result. And you can see that in our earlier data, the percent of people who had a positive test result and reported loss of sense of taste or smell was 58%. But in the more recent data, that's dropped down to 28%. And for sensitive skin, similarly, it was 39% earlier on, and now it's dropped down to 10%. So overall, there seems to be this this trend for the number of people who actually have these classic symptoms to be decreasing as the number of people that you test widens, and as probably the severeness of the illness of the people who are tested is decreasing. I did a quick look to see if the symptoms that are more associated with having COVID-19 are also associated with having more symptoms reported in general. So what's plotted here on the x-axis is the odds ratio for symptoms being enriched in people who had a positive COVID-19 test versus a negative test result. And in the y-axis is the total number of symptoms reported on average by each person who reported that particular symptom. And so there isn't a strong correlation here. There isn't a great take-home story from this, but there is perhaps a nominal association for symptoms that are reported concordant with other symptoms to also be enriched in COVID-19 cases. I should say these are all very preliminary results. None of this is published. These are more exploratory analyses that I've been doing that I would like to see replicated in additional data sets. But in conclusion, I think when we're looking at the symptoms that are associated with COVID-19 positivity, there are a lot of complications that we're going to need to think about. So we need to be taking into account how sick the person is overall. Is it that some of these more classic symptoms, are they associated with COVID-19 specifically, or are they only associated in the more severe COVID-19 cases and not so much in the less severe COVID-19 cases? Or are some of them just associated with being severely sick in general as opposed to being specific to COVID-19. These are all things that should be teased apart. And of course, as time goes on, I think we'll also be able to pick apart better the differences between people and their symptoms by ethnicity, by sex, by age, and eventually by genetics. And that will be extremely interesting when we get to that. I'd like to acknowledge multiple people from Helix who helped with this work. And if there's questions, I'll take time, or we can go to the next speaker.
Thank you, Liz, so much. You do have questions. If you could answer them by typing in the question and answer box in the interest of time, that would be great. I noticed you already have a few. And also the other speakers, you have questions directed to you. So if Cristina Cristina could take a look, and maybe Valentina too. So that my next pleasure is to introduce Patrick Deelen. So Patrick, please share your screen. He comes to us from the University Medical Center in Groningen. So Patrick, if you're there, can you please share your screen?
Yet, and I'm unmuted now. So can you guys hear me?
Go ahead. We can hear you, and we can see your screen.
Great, thank you. So I'm Patrick Deelen. I am a postdoctoral researcher at the University Medical Center in Groningen, and I would like to talk to you about our Lifelines biobank and the COVID-19 research that we're doing with that. So the Lifelines cohort, it's a cohort of 160,000 individuals. And we started sending out questionnaires to these people at the start of the pandemic. And actually several things that we try to do here. So we try to identify environmental factors influencing COVID-19 and then both susceptibility as severity. That's the same thing we'll do for genetic differences. And aside from that, we will also measure the effect of the pandemic on mental well-being and psychological welfare, and all kinds of different social aspects. So as I said, we started in the beginning of the pandemic. On March 30th, we sent out the first questionnaire. And we've been doing that in the beginning weekly, and now we're sending out bi-weekly questionnaires. So up to now, 250,000 questionnaires have been filled in. And each round we get about 40,000 to 50,000 respondees. Up to now, we have 116 positive COVID-19 tests and another 811 COVID-19 diagnoses by a medical doctor. Although one caveat here is that apparently medical doctors are not very good at that, because if a week later people have been tested, they're more often negative than they are positive. So we should be careful interpreting those. So a bit of background information on the questionnaires. So we have all kinds of different things. So there are more social demographics, like the number of housemates that you have, what kind of job you do, whether or not you're working at home or from afar. From the Lifelines cohort, we know already most of the chronic diseases people have, but just to make sure that it was up to date, we asked this again. We have all kinds of COVID-19 related questions, so whether or not people have been tested, what the outcome is, if it's a diagnosis by a doctor, hospital admissions, have they been on a ventilator, or was it more basic support. We have health questions like the list described from Helix, so we ask for fever, cough, headache, and many others as well. And based on the recommendation of Liz, we also started asking sensitive skin. Every week we ask what kind of drugs people have been using the past week, which we are converting to ATC codes. We have a mental health questionnaire with the mini questions, and then it's a well-being, number of social contacts people had, the frailty index, especially for older people, a kid screen to see how the kids are handling it, and we now also started asking for the vaccination status. So both childhood vaccinations as more recent flu shots or travel vaccinations. And these are all things, of course, we then want to relate to the COVID-19 status of people. So one of the things we have been doing, and this is in Dutch, is just to inform the general public. So we made a corona barometer. We're working on an English version as well, where we show the public and the participants some statistics on what we're doing. And we allow people to visualize this over time, allowing also the comparison of two different measurements. And then, of course, one of the things we have also been working on, can we predict maybe this COVID-19 so that we can run GWASs on this, even though testing has been limited. And then we can look at things like loss of taste or smell. And then we actually had this on a 5-point scale. And we see that the positive cases are very strongly enriched for loss of taste or smell, but by far not all people. So there's actually about a little less than half of the people that don't at least describe this at all. And you also see a very striking difference between actually a positive test and a doctor's diagnosis, because apparently the doctors don't judge whether or not people have loss of taste and smell, so that they don't select on that. And the same, actually a similar figure is for fever and many of the other traits. One thing that we do notice when you try to make predictions using these phenotypes, that for instance neurological diseases, that they are strongly enriched for loss of taste and smell. And the same holds for lung diseases and asthma. So for each of these phenotypes, ideally, if you have the power, you should correct for that as well when you try to predict your positive COVID-19 cases. So as I said, this testing is limited, so I won't actually repeat this model because Liz just described it, but this is just one model to predict your cases so that you can then do GWASs on that. And we've been doing that with the COVID-19 Host Genetics Initiative. And this is actually a very large initiative with over 180 curated studies. So these are all studies that studying the interaction between genetics and COVID-19, either susceptibility or severity. And so far, the first GWASs have been done on about 10 of these studies, and these are mostly biobanks that already had genotypes available. But many of these other studies are now focusing on getting genetics in or getting their data in, so that we hope to grow very far beyond these 10 contributing studies. They have different types of analysis described. So the first 4 are more on severity. Then we have 2 that are aimed at susceptibility. And then it's called here the flu symptoms, which is aimed at the predicted COVID-19 based on symptoms. So just to illustrate the results here, the first one is whether or not COVID-19 cases has been hospitalized. And now I take you guys back 15 years because, well, doing a GWAS on little over 1,000 samples is of course a bit old-fashioned. And as expected, we don't really see a signal here, at least no significant hits. The COVID-19 infection versus population controls thus is yielding the first barely genome-wide significant hits, and some interesting peaks are starting to pop up. And the last one we did so far is the analysis 7 is the predicted COVID-19, so using the spectrum model. And there actually we also already see some signal, even though there are fewer samples. We did some different analysis on this data, one of which is downstreamer. And this is a method that we are developing in-house already for other GWASs. And maybe some of the GWAS people know the PICT, which is used to predict pathways relevant for GWASs. Now we are now using downstreamer to predict both pathways, but also the core genes, so the genes that are really essential for the development of a disease, which are actually very often Mendelian disease genes. And we do this by using the full GWAS summary statistics. Now, interestingly, the analysis on the predicted COVID-19, which is also on which the predictions are very importantly driven by loss of taste and smell, is that we pick up signals from macroautophagy and phagophore assembly sites. Now, the first one is, of course, a very general pathway, but the second one alludes already to the possible mechanism of a virus, because that's, of course, done by macroautophagy at these phagophore assembly sites. And these results are actually, even though we didn't have any genome-wide significant SNPs, these enrichment analyses are genome-wide significant. What is not significant is the tissues that are enriched, but the top 2 actually do make a lot of sense. So that is lung and this is mucosa tissue. So I do think it's worthwhile to mention that. The other heavily underpowered GWAS that we did was this analysis 2, which was hospital admitted versus non-hospital admitted cases. These results are not bonferroni significant, but they're only very little below, so they're definitely within the FTR 5% range. And here we find this bitter taste receptor activity and also these pheromone receptors. So at first glance, this may not make a lot of sense, but given that you have this COVID-19 loss of taste and smell, which is very common, to some extent that does make sense that you then have taste receptors popping up. But how we should place this within the activity of the virus, we really don't know at this stage yet. Just to show you as well, we're doing also EQTL analysis, so seeing if the variants given COVID that are yielding COVID-19 susceptibility are also regulating gene expression. We have two overlapping signals. One of them is in an IGLV region, which is likely a false positive due to the many different homologs of this gene. And the other one is in the mitochondrial tRNA gene. So once again, we don't know how to explain this, but this is just to show what kinds of analyses are already going on. And then lastly, we're also using single cell EQTL data to look in the different cell types, what kind of EQTLs we can find there. So this is of course a very young consortium because this disease didn't exist half a year ago. So the analysis plans are being completely rewritten, although the essentials are exactly the same. But this is really in flux and ongoing. So this is likely to change a few more times. We're also trying to improve the COVID-19 prediction. So we trained a model in our Lifelines data, but it might be a bit too population specific because when validating in the Helix data, it actually performed very similar to the previously published models. But these are things that we hope to improve, that we can do better GWASs here. And we're within the Netherlands are also setting up larger collaborations with different biobanks, so we try to harmonize our data as much as possible. This is of course a very big effort, so there are many people and collaborators that I would like to thank. And that was what I wanted to share today.
Patrick, thank you so much. I direct you to the question and answer box. You seem to be amassing quite a few questions, so I'd ask you to go ahead and type your answers to people that have questions. And our next speaker will be Diego Restrepo, coming to us from the University of Colorado. So Diego, can you please go ahead and get started and share your screen?
Yeah. You see my screen?
I can see your screen and I can hear you.
Great. Well, I'd like to thank the organizers, and it's nice to be at Monell, even if it's only virtually. And I'd like to talk to you about the cause of loss of smell. So what's happening in the olfactory epithelium? And this is a study with transcriptomics that we did. The main authors are Danae Baxter and Eric Larson, but we have a large group of people, multidisciplinary. So let me go into the talk. So first of all, Bob Data gave a great talk last week about the entry of the virus of the SARS-CoV-2 into the host. And this is basically what they described was there's a need for interaction of ACE2 with the spike protein. And also there's a protease that's important in this process. And they found that this ACE2 is expressed in the sustentacular cells. And I'd like to give you a different perspective, something that's complementary, which is what happens in the olfactory epithelium. On the right side, you see this picture from Durante. And what you see is that this is a fairly complex tissue. And it has both, of course, the sensory neurons, the olfactory sensory neurons, but it also has other cells, and some of these are these sentinel cells, the microvillus cells that I'll talk about in a moment. And also, of course, you have immune system cells over here. So, and the point I would like to make is that once you have the infection, this is likely dynamic and things are changing. And so let me just go to the next slide. So this is the work from Bob Data, and there's also the Deelen paper. And what they found is that when you look at the receptor for the virus, this ACE2, it's found in the sustentacular cells, but it's found in a fairly low percent of the cells. And this is a transcriptomic analysis. They also found that TMPRSS2, which is basically a serine protease, which is important for the entry of the virus. And this is complementary work from Andy Lane in Hopkins, and this is with the antibody showing that, yes, it is in the sustentacular cells. The interesting thing is that there's been some reports, and this one is from Ziegler et al., where they have found that when they infect in vitro, they actually get a change in ACE2 expression. So in this one, it was done from human donors, and this is nasal scrapings, and they found that if they use interferons, they actually get an increase in ACE2. So this is a dynamic process. So what we'd like to look at here is to look at TRPM5 expressing cells. And I'll go in a moment I'll show you the cells, but the reason why we're interested in the TRPM5 expressing cells is because in many parts of in the epithelium, including the gut, this one I'm showing from actually somebody who gave a talk 2 weeks ago, of a monkey. It's these tough cells in the intestine. And they started type 2 inflammatory reaction to different challenges like bacteria, hemolymph, and also virus in this case. That's also been shown in the lung by Noam Cohen. And the question is, what's happening in the olfactory epithelium? What do they express? So we looked at the transcriptome. Here's one of the first, this was with Bob Margolskee, one of the first studies of the TRPM5 expressing cells in the olfactory epithelium. This was done with a TRPM5 GFP mouse, eGFP mouse, in fact. And what we found was two classes of cells. These are these tuft-like cells. These are these microvillus cells, we call them. And so they're epithelial cells. And you also have a subset of olfactory sensory neurons. This was work from Wei-Hong Ling, and she's followed up on this, and she's found some really interesting facts, which are that they respond to ATP and chemicals, and usually chemicals at high concentrations, so mild irritants, basically. They release acetylcholine. They actually have a paracrine effect on sustentacular cells, which is interesting. And they also activate proliferation and apoptosis of OSNs, so the death of OSNs, active death. And I'll come back to that in a moment. This is just showing some studies that they did with changes in calcium in the microvillar cells. So what we did was to do, so these cells are a small percent of the population. So what we did is we actually used FACS, fluorescence activated cell sorting. We isolated the cells from the olfactory epithelium in mouse, in fact. And this is where Paul Feinstein comes in. He made a mouse that expresses M-cherry in the mature neurons. So this is an OMP M-cherry mouse. And then we crossed it with the TRPM5. And you see two cells here. This is an olfactory sensory neuron that's TRPM5 positive, and that's one that's negative. We sorted the cells, and then we took, this is the y-axis is the M-cherry axis, so this is the OMP axis. Mature neurons up here, immature neurons and everything else is down here. This is the eGFP, so this is basically the microvillar cells that are OMP negative, and this is the subset of olfactory neurons that are positive. These are small populations, so this is a really nice way of separating them. You have about 1 million cells here, and you have about 1,000 cells here, and maybe 3,000, 4,000 here. What we did then is do RNA-seq, and the results were pretty interesting. So first I'm going to go, since I'm in the Rocky Mountains at the 14er, so at 1,000-foot view, really 14,000-foot view. And so I'm going to look at only the 10 top differences between, in this case, the OSNs. I have to move these over. Okay, I'm going to call the ones that are EGFP negatives, so the olfactory sensory neurons that are EGFP negative. These are the ones that are EGFP positive. And the 1,000-foot view is basically looking at only the 10 top genes. So this is pretty interesting. What we find here is that they're very different in a subset of olfactory receptors. So there's a subset of olfactory receptors that are very low in the EGFP positive ones. Something else that's really interesting, this is something, a protein that Tim McClintock studied. The S100A5 is a protein that tells you that the receptors are responding to odors, and they're actually quite silent, the EGFP positive ones. So that's what you find here. The other thing that you find in the EGFP positive ones that you expect is that they're positive for GFP. That makes a lot of sense. That they're positive for TRPM5, which makes sense. And by the way, this is full-length TRPM5. I will not show the data. But they're also positive in things like ESPN, which is basically for microvilli, which is not the case in the negative ones, the EGFP negative ones. And they're positive for other things like these two are involved in peripheral injury. So that's already interesting. This just shows all of the olfactory receptors that are different, and only 25 are positive in the EGFP plus, and the rest of them, 500 and something, are negative in these cells. The rest of the receptors are not significant either way. Now we look at the microvillar cells. So these are called MVC EGFP plus. So they're here on the left side, versus the olfactory sensory neurons, the EGFP minus and the EGFP plus. And what we find again is fairly interesting. And what we have is you have a very large difference in synaptic proteins like SNAP25, which are positive in the sensory neurons. That makes a lot of sense. S100A5, that's positive in these. And then the things that you find are higher in the MVCs are like POU2F3, which is also SKIN1A, which is a transcription factor for the production of these cells, and other ones that are like TRPM5, and you'll see in a moment, you see the sweet taste pathway is positive in these cells as well. So that's the MVCs. So that makes a lot of sense. That's what you find in the staph cells, the sentinels. On the olfactory neurons, you find on the other side the olfactory transduction pathway. So that makes a lot of sense. Now, the thing that was really interesting was that when we went to the gene ontology and we looked at the overall, we found large differences in inflammatory, immune, and viral-related genes for both the olfactory sensory neurons that were EGFP positive and the microvillar cells compared to everybody else. And in fact, you find increases in IL-25, which makes a lot of sense for a tough-like cell, and also in ALOX5 leukotrienes, basically, production of leukotrienes. The other thing is that one of the large differences was in virally related gene expression. And I'm not going to go into details, but there's a large number of proteins, some that are important in adhesion, in entry, some that are important in replication, but you don't form a very clear story of what these are doing. Some are important for viral defense, and some are actually important for the viral entry. The final thing, since I'm coming close to the end, is that we did some infection, in this case not SARS-CoV-2, but we did HSV1, basically cold sores, that does infect mice as well as humans, of course. And what we found, which is pretty interesting, is we found, this is, by the way, on purpose, only a 5-day infection, and it's an ARES infection, and it's a high titer infection. And what we find is a decrease in the olfactory sensory neurons, both the GFP plus and the GFP minus, and no change, no statistical change in MVCs. In fact, it looks like they might be going up. And that's interesting, going back to Wei Hong Lin's finding that when she stimulates with irritants, she finds an increase in caspase-3 apoptosis. So in the olfactory sensory neurons, the loss of olfactory sensory neurons in the anterior part of the epithelium. And she does not find that in the skin 1A, that POUF2, which is basically, they don't have these microvillar cells. So in the final conclusion is basically that these cells, the microvillar cells and the OSNZ GFP plus, are fairly, and the GFP minus are fairly distinct. They actually share, each of those share something, but they're also different because of many different transcription factors. The microvillar cells and the OSNZ GFP plus display enhanced expression of immune, inflammatory, and viral-related transcripts. To what extent does recognition of SARS-CoV-2 by 3.5 expressing cells participate in smell loss? It's not clear right now. Is the immune system involved? It could be, and that's something to do in the future. And finally, I just wanted to show everybody here, and basically Danae is over here, and Eric, I could only find your picture looking into the microscope, I'm sorry, and then the rest of the team. So thank you very much, and I'll be glad to answer questions either by typing or answering if there's any time.
Diego, thank you so much. I'll direct you to the question and answer panel to see if you have questions. And while we're doing that, this leads us to our last speaker of the day, and I'd like to invite Chrissi Kelly to be sharing her screen. Chrissi is going to be giving us her experience of what it's like to deal with a lot of the COVID patients who have anosmia and some of the things that she's been on the front lines to observe. So Chrissi, can you go ahead and share your screen? Good, we can hear you. We can see your screen, Chrissi, go ahead. You're muted, so please take yourself off mute.
There we go. So,
yeah.
Thank you very much, Danny. It is a pleasure to be here. And I have to say, you know, despite the pandemic, one of the great advantages has been being able to sort of work with you so often. So it's great to be here. I just wanted to, so let me just move on to the next screen. And I should start by saying that I'm a patient advocate. I am the founder of a charity called AbScent, and we are registered in England and Wales. I've been running it on a sort of ad hoc basis now since 2015. And in 2019, we got registered charity status. So, and we provide through our Facebook groups and the website, we provide support, education, and practical advice for anyone that comes to us with a smell disorder. Of course, we have many people who do not have post-viral smell loss, but other kinds of smell loss. So we're sort of, we welcome everyone. I started out in 2015 with the sort of original AbScent Facebook group. And then in February, I started receiving tweets and getting messages specifically from Italy saying that that this seemed to be a feature of COVID-19. And around the 13th of March, I took a look at my statistics and saw that we were rapidly growing. Let me take you to the next slide. So there I was sort of going along quietly with 1% growth in my membership per week. That was a completely straight line, very little variation. And then suddenly around the 13th of March, things started moving. And then we started getting, the curve started to go straight up. On the 23rd of March, we were completely overwhelmed in the original Facebook group. And I would describe it like this. We were enjoying multiple conversations every day about things that were on everyone's mind, regardless of whether it was post-viral or something else. And then suddenly the doors burst open and in piled all these people who had COVID-19 smell loss. And they were very, very anxious, wanting to talk about the virus. And pretty soon it became obvious that we weren't going to be able to have both the COVID-19 group and the original AbScent group together in one space. So that idea came to me at 10:30 at night. I quickly set up a Facebook group called AbScent COVID-19. And then when I woke up in the morning, there were 100 people. And by the end of the week, there were 560 members. So it grew very quickly. Today, we've got over 2,200 members. And it's a very, very active group. So this is what's happened within the group since we started the COVID-19 group. We've had 1,725 conversation threads, and that just starts. Anyone can come and ask a question, and then it's then posted. We have multiple questions every day. And of course, every question inspires comments. So, you might have a question or a post that has 50 comments. So it's a very, very active group. And you can see how many conversations, comments, and reactions we've had in the time that the Facebook group has been running. So what can we learn about this? I have to start by saying that I didn't have the original AbScent Facebook going for very long before I realized that it was a very interesting source of information. Just to sit there and watch and listen to what people were talking about and what was on their minds, that is, of course, was the basis of the genesis of the parosmia study I'm doing with Dr. Jane Parker at the University of Reading. If it hadn't been for the comments that kept coming up about parosmia and what kind of foods trigger parosmia, I would never have sort of put that together and then sort of gone. I gave a talk in Amsterdam last March, and Jane was in the audience, and that's how we came together. But it really is, if you look and listen to what's happening, you do start to see some very interesting trends. And so as the person who is in charge of the COVID-19 Facebook group, I'm in a very privileged position to watch in real time what is happening to the patients who are coming into the group. It's sort of like saying, okay, it's going to be a marathon. We're going to say go all at once. Maybe not everyone leaves at the starting gate at the same time, but that's the sort of bulk of the people sort of pass through. And then you get to watch as some people arrive quickly, some people take longer, some people decide to walk instead of run. So you see in real time what kind of outcomes are we seeing here? What kind of trends are we seeing? Are they different from what we would normally expect with post-viral anosmia? And that's been very interesting. So just a couple of things that I'd like to tell you about right now. The first one, of course, it comes as no surprise that parosmia is trending as a subject in the Facebook group. We're getting more and more of that. And in fact, I was also contacted yesterday by the Iranian ENT, who was the lead author on the 10,000 person study there, to say, we've just got masses of people now with parosmia, what are we going to do? So I think that is something that's really coming to the fore now. More details about that, whether it's going to follow the same timeline as normally with post-viral anosmia, we just don't know now, but it's very interesting what they report. They report the same kind of, they use the same descriptors when talking about the distortions to their sense of smell, the same kinds of foods that are triggers that we always anticipated. So no reason to suspect that this parosmia is any different from the parosmia that we knew before COVID-19. But it is nonetheless interesting to see that that is very much a trend now. Valentina mentioned the results of GCCR001 and the interest in chemesthesis as well as gustation that are these changes that are being reported. So we hear a lot about that in the COVID-19 group also. But one thing that interests me when people talk about these kind of chemesthetic sensations is that they're not always the same. So for instance, people will report that they can't smell bleach, for instance, but they can smell things that are menthol. Sometimes they can not taste chili. And again, we'd have to wait for further studies to find out how these variables all stick together. But there are reports of all different kinds of loss, both the kind that are detected in the nose through orthonasal olfaction, as well as those that are available in the mouth, the receptors in the mouth that don't seem to be functioning normally. So that is an interesting one, and I must say that in my time managing the AbScent, the regular AbScent Facebook group, I don't see that much in the way of comment about loss of gustation or in fact chemesthesis. So this does seem to be a new trend and something that we've been seeing in the COVID-19 group. Something that people discuss all the time is fluctuation. And it's interesting that the COVID-19 group is very, very specific about how much of their sense of smell they have available to them. And this is something I've never seen before. They'll say, oh, I've got 10% of my smell, 60%, 90%. And these are ways of describing sense of smell that I've never seen before in the original AbScent group. And what they'll do is they'll come in and they'll say, today I have zero sense of smell. I woke up, I have zero sense of smell. I'm back to where I started from. It's very depressing. And then they'll report that this then comes back. It might come back a couple of hours later or the next day, but there seems to be really just a lot of fluctuation in their ability to smell. Whether that's reflected with gustation and chemesthesis, I'm not sure right now, but I do know that as far as smell is concerned, that is definitely a feature. And of course, patients find this extremely frustrating. The last trend that I wanted to describe is this one about burning and dryness in the nostrils. And I think this is something that is also described often. And this is something that people find very troubling, very painful. This feeling of excessive openness in the nasal passages, which is associated with pain behind the bridge of the nose. This is something that also comes and goes over, so people will feel they've recovered, and then that time will go by, they'll feel quite normal, and then they'll get that again. So those are just a couple of the trends. Naturally, comments in a Facebook group like this are difficult to quantify. There is a data mining that can be done, but we haven't yet got that far. So I would say that just reading through what's happening in the Facebook group is perhaps interesting for anyone who is working on this problem, just to see what people are saying. I find it very interesting. And lastly, I would say that it doesn't often happen that people will turn up with their complaint of having lost their sense of smell so soon after it happens. Normally in the Facebook group, in the old AbScent Facebook group, people might come in, join us, you know, after 3 months had gone by, 6 months had gone by. Many times they didn't know the word for anosmia. That has stopped. Everyone knows that smell loss is associated with COVID-19. Even when I went walking the dog the other night, a neighbor who I do not know very well, she waved to me. She said, "Oh, aren't you working on something to do with anosmia?" So suddenly this word is now with us. And as a patient advocate who has been trying for a long time to work out how to raise awareness, I can say that we are now there. I mean, it's as awful as the pandemic is, it's changed the landscape for us. We now have more, there's more awareness about the problems associated with smell loss. And therefore, because there's this greater awareness, the affected patients who are finding out about it are going straight to Facebook, looking up groups that might help them. And they're coming in saying things like, "I lost my sense of smell yesterday." Now that's incredible. And so those patients also would be a very interesting source of information to capture them so early on to see exactly what's happening with them. And for that reason, I recognize this, the responsibility of being in charge of a group of people like this. So I do try my best, given my resources, to monitor it. I do advertise surveys that are hopefully of benefit to the patients in the end effect, of course, but also to the kind of research institutions that contact me and ask me for assistance in reaching patients. So I consider that as a very important part of my outreach. And otherwise, I guess that is really what I have to say. I would like to say thank you very much to Monell for inviting me again and helping me do my job of looking after patients. So that's it, really. Thank you.
Chrissi, thank you so much. And I'd just like to thank all the speakers for being with us today, and of course, all of our panelists. And particular shout out to Katie Long and Alyssa Treff and Karen Krieger, who did a lot of the organization for this event. We're going to take a little minute to have a stretch. This formally ends our program and presentation today, but I invite all the speakers and the panelists who want to, and the attendees, to stay on the line.